the series. Okay. I'm just going to mute Linda for now. She's got some background noise. Welcome everyone to the online MOOC for learning AMR, screening and theory. This is the first of those MOOCs, and this is in Bacteriology 101 and sort of antimicrobial resistance and how it sort of propagates. This is brought to you by the Microbiology Society, Jogel, Kamasi Hive, and MUST. And we've got a guest here today, Professor Courage K.S. Saba as well, and myself, who will be leading the lecture. So as a sort of introduction to the course, the online MOOCs are all hosted at this link here, which is Jogel. It's in a challenge. And you'll find the resources that we've used and the lecture slides, as well as the recordings, all available here. And this is sort of the current plan of the MOOCs. So today we're learning about bacteriology, microbiology, and antimicrobial resistance. Next week, we'll be doing techniques for screening and identification of those microbes. The week after, sensitivity testing and antimicrobial susceptibility. And then we've got AMR stewardship, digital mapping using Microbes, which is created by Harry here, who's in the call with us. And then in late October, we'll be announcing who is joining us for the practical side of things, and we'll be giving certificates of attendance. And also later on in the program, it's not only for those who have attended, but we'll be hosting a grant review round and an opportunity to get some money to do your own antimicrobial project. It's nice to see you all arriving in the chat as well. So this is brought to you by Professor Courage K.S. Saba, who's with us today, and he can talk when he wants. So we've got Linda, Gamily, Joy, Harry, myself, Chris, and John Moat, who is a guest speaker on the third MOOC. Got a range of different expertise from management, specialization, to sort of masters in antimicrobials, and also a professor, of course. I've talked you through the MOOC summary. But so today we're going to be talking about microbiology and sort of more into the bacteriology side of things. So microbes could be viruses, nematodes, insects, fungi, archaea, but we'll be looking at bacteria. And within those bacteria, there's a huge array of different morphologies. And we'll be looking at sort of a specific subset of these, and they all have different niches, of course. So what is a bacteria? A bacteria is a single-celled organism, a prokaryote. And inside this single-celled organism, we've got a nucleosome, which encodes the DNA. And then outside that, among loads of other processes that allow it to replicate and divide, we've got the cell envelope, peptidoglycan, and sort of a mucus layer, as well as extra things like flagella that allow it to swim around as well. And we're actually going to look more closely today, specifically at this type of bacteria. And when bacteria were first sort of looked at, there were different ways of identifying bacteria. And of course, there's morphologies, the different shapes of bacteria, the different sizes, but also there's the gram stain test. And effectively, there's this crystal violet dye that you can add to bacteria. And some of them, they turn a dark violet shade, and others, they instead retain this pink if you counterstain them. And so gram-negative do not stain with crystal violet, and gram-positive do. And originally, it wasn't really known what this was. But now we know that these two bacteria actually are classed by the sort of cell envelope they have around the cell. So that's this thing here that goes around the cell. And gram-negatives, they have two layers, and whereas gram-positives have one. And so the gram staining, actually, effectively, it's crystals growing and being trapped in this layer here. And this layer here is chains of sugars that are connected by peptides. And in gram-positives, this is reaching outwards, and it's quite large. and it goes away from the lipid bilayer here. Whereas in gram-negatives, and what makes them so powerful against antibiotics most of the time, is that we have a lipid layer that actually protects this layer here. So this layer, which gives the bacteria its shape, which is quite solid, that it attaches to that and protects it, in addition to the normal layers. And then we have aerobic versus anaerobic bacteria as well. So if we have all around us, we have different environments. Anaerobic essentially means high in oxygen concentration, and anaerobic means very, very low or not tolerant to oxygen at all, because it can damage them. And so if you have an obligate anaerobe, it could be in our gut, for example. And if it's an obligate aerobe, it could require high amounts of oxygen to sustain a viable lifestyle. And of course, there's also bacteria between. And what's interesting is when you're working on anaerobic bacteria, you need it's an anaerobic cabinet, so to prevent all the oxygen from going inside. Now, the reason why we're focusing on gram-negative bacteria, and that is those bacteria with 2 layers, is that some of the most relevant bacteria in AMR are those that are gram-negative. So we have Clostridium difficile, which is not gram-negative at all, but is anaerobic, and much more. We have these gram-negative bacteria, so salmonella, E. coli. These are bacteria that have got high resistance profiles, and that's because these extra layers give them a sort of protection against the antibiotics that can affect the peptidoglycan, which gives them the cell shape. And then in addition to that, we have the mycobacterium. So we have gram-negative, gram-positive, and in addition, we have the mycobacterium, which is a bit different altogether, and is even more powerful in some cases. And these bacteria, they have not just the outer membrane, inner membrane, and peptidoglycan, but instead they have these extra sugars and ionogalactans, and also extra lipids that sort of protect the bacteria to an even greater extent, and it's all covalently linked. And what's even interesting is that these layers are so hard to separate that it's not very well understood even now. But these actually don't show up in gram stain as purple, because the peptidoglycan, which would hold the crystals, can't be reached. But in the same case, they're not gram-negative, as they don't have this extra layer. It's much different. So how do these bacteria grow? So this is the first module, so we're just going from the very, very basics. The DNA, which is a sort of set of nucleotides, adenine, thymine, cytosine, guanine, these can code for amino acids. And effectively, we have this double helix here, and every 3 amino acids we get, every 3 nucleotides can code for a different amino acid, and that's here. So we've got 20 typical natural amino acid structures. And then effectively, so we have ribosomes, which are floating always freely through the cell, so these ribosomes here. And in bacteria, they attach to the nucleic acids, and they allow for coding of amino acid chains. And so you get chains of beads that build proteins. And these chains of beads interact with one another, and they form a 3D sort of structure, and the infinite complexities and forms, which can allow for the sort of complexity that we see in life and bacteria itself. And they also allow for the building of these bacteria, and they can do many things. So there are some proteins that make the cell wall, and we're going to be talking about these because they're especially relevant in antimicrobials. So we have proteins that build the cell wall by stitching sugars together, attaching amino acids to them, and building these strands here. They're called PVPs, or sense complexes. And we have proteins that can move lipids from the inner membrane to the outer membrane as well. And in addition to that, we have lots of other roles, but these are quite interesting in this case. And antibiotics, we want to use antibiotics to kill bacteria, and that's because lots of bacteria like to kill us or give us awful diseases. And so there's many different ways that antibiotics do this. And these are normally produced by other bacteria that might want to kill bacteria around them in a competition kind of wise manner, or other fungus or around the world. And so we have some that inhibit the production of proteins. They inhibit the ability for these ribosomes to actually create chains of amino acids, for example. We have those that stop the cell wall that we just talked about from being produced. They block the proteins from creating the cell wall. We have antibiotics that actually destroy or make holes in the layers as well. So especially in gram negatives, where we've got 2 layers, this is especially important. We've also got nucleic acid synthesis. So every now and then we need to make a new copy of DNA to replicate and create 2 cells instead of 1 or more. And there are some antibiotics that can block this process as well. So they can bind to the proteins. Sometimes it's permanently. And we're going to be talking more about beta-lactams in a moment. This is just a slide. If you want to read through it later, this will be available later. So the classic example of an antibiotic is penicillin. And penicillin is produced by Penicillium chrysanogen, and it's just this little chemical here. And penicillin is part of a group of beta-lactam type chemicals with a variable group that we can add different things to, and that we get like a range of different bacillins, for example, antibiotics that are derived from this. And effectively what they do is the proteins I talked about before, which produce the peptidoglycan in gram negatives and gram positives, they can be inhibited by beta-lactams. So effectively the beta-lactam mimics the peptidoglycan or lipid tube or sugar plus peptide group, and stops these from performing their task of joining these amino acids which are attached to sugars together, and therefore stop the mesh being formed. And so that's what penicillin is doing, and that's how it sort of works and allows the cell wall to, instead of being in a sort of a growth phase, it actually is stopped from growing and it becomes unstable and the whole system breaks apart and the cells sort of explode or inhibits growth. And so to reiterate how this happens is we've got sugars that are being put together in the cytoplasm here and being added to, we've got little amino acids that are added to it by a series of MER enzymes. That's then flipped with using a fat here, and then into the periplasm and joined together by transglycosylation. And then it's the transpeptidase action of joining these amino acids together in these chains into a mesh that is stopped by the penicillin. But the problem is that, as is always the case in life, there's mutation and evolution that tries to evolve away from these processes. And we get resistance. So bacteria, they can change their wall proteins. So instead of it binding, maybe it doesn't bind. They can modify the drug target. They can create an efflux pump. So a lot of the time, maybe they do actually have an effect, but the antibiotic can be pumped in and then actually be pumped out, straight back out again. So they have a huge amount of pumps that are actually making them resistant to many different types of antibiotics, which is a big problem. And then also we've got drug inactivating enzymes, which is even more a problem than actually something to think about with PVPs. And so what happens is we get these resistant bacteria, which have a small amount of resistance, or which is resistant unlike all of its friends. And we get an increase in population of these bacteria because the rest are killed. And so over time, these little red bacteria increase in density until the entire population is resistant. But it's not just this, and you can see this here in action. So this, the yellow or more opaque parts here on this agar petri dish, we've got bands of inhibition or death around these antibiotic discs. And there is like different concentrations at which they kill. But in some cases, you'll get small little colonies, which says, oop, there's now a resistant colony to that. And over time, that'll increase until the entire population is resistant, for example. And this is all fine and good, but actually, if we were always discovering antibiotics, it wouldn't be that much of a problem. But the problem is that penicillin, which was one of the first antibiotics to be discovered, there was a huge surge in antibiotics being discovered after this, because we realized that fungi and lots of other natural products were antibiotic and could kill bacteria. But then from about 1990, there's been a discovery void, and that there's no new antibiotics that can create a new family of antibiotics. And that means that we're in trouble, because over time, there's going to be a resistance to all of these antibiotics. So here's an example of one type of resistance, and that's the Betalactamase. So the Betalactamase is this sort of protein here. And we talked before about penicillin and its beta-lactam ring and how it attaches to PVPs, and how that can stop the cell wall from being effectively created. But what happens here is the beta-lactamase, which existed naturally in some bacteria already, it takes the intact antibiotic and it destroys it. It destroys the beta-lactam ring. And what's more is that these pieces of DNA aren't just shared in one species, but can actually be transferred between species. And that's through, a lot of the time, conjugation. So we get pieces of DNA which are in their nucleosome, and there's small exchanges that can occur over time with plasmids, but can also have elements of plasmids themselves. And these plasmids, which contain DNA, which contain the information to create proteins, such as the beta-lactam and the beta-lactamase rather, can be received by another species. And so this now is resistant also to the beta-lactams, for example. And this isn't just beta-lactamases, this is many other forms of resistance as well. So what we have is we have huge clades now, huge numbers of species that have become resistant to bacteria. And it's a big problem. But there are alternatives other than chemicals. We can also use phage therapy, for example. So here's another type of antibiotics. So a phage is a small virus that can kill bacteria, and it replicates within them. So what we get is we get the absorption of the phage. It takes its DNA that it holds within it, puts it into the bacteria. The bacteria, it does depend on the species of phage, of course. The bacteria then uses the DNA that's put in there and creates little pieces of virus, and then it assembles itself, and then it lyses the cells, and then you basically you've killed bacteria. So there are some countries that actually have phage therapy as a major form of antibiotics. So this could be the future. And what's interesting about phage therapy is phage is also evolving. So it's not like with antibiotics, where we've got an antibiotic which is a static chemical, but in fact we can have a range of phages. But there is one disadvantage in phages, and phages tend to be very host specific. So this phage here will have a protein that binds a protein of the bacteria, and that's required. And so these are species specific and require lots of research. And then we have a sort of a hero bacteria, which I love. It's called Del Vibrio. Del Vibrio bacteriovirus is sort of used in lots of studies, and it's a smaller bacteria with a big flagellum here, and it attacks gram-negative species of bacteria. That is those species with two membrane layers, as we said before, and it's evolved to make and puncture small holes into the bacteria, and it lives inside. So it goes through, creates a small place for it to live in the periplasm, and then it eats the inside of the bacteria and grows like a sausage, septates into lots of little bacteria again, and then finally escapes and lyses. And what's interesting about Del Vibrio is there isn't just one species, it can actually kill many, many species. And so there's been studies in both phage therapy and in Del Vibrio. So in the future, it might be that rather than using big chemical plants or creating or discovering new chemicals, it might be that more locally we can use Del Vibrio or phage therapy to solve the antibiotic crisis. And what's important about all this is you actually, to know what phage or Delvibrio or antibiotic to use. You have to identify the bacteria. And so we're going to be talking about that next week. And so in summary, we have gram-negative and positive bacteria, the cell wall, DNA codes for amino acids, of course, I'm sorry about that, the antibiotics, mutations and the rise of antibiotic resistance, and also alternatives to chemicals. So that's the first half of this lecture, and the next half will be hosted by Professor K.S. Saba. But now we've got a little question session. I can only take about 3 questions. So I'm just going to look in the chat if there's any questions in the Q&A or anything. If you do have any questions, please put it in the chat, and I'll be happy to unmute you and ask. I can't see any questions yet, so I'm just going to let Courage start with his part, I think.
Yeah, thank you very much, Chris.
So there's a question.
Oh, okay.
Can I continue?
Oh, I think I'll answer the first question. So, Abdul, why haven't we found new antibiotics of late since the 1990s? And I think that the reason why we haven't found antibiotics since the 1990s is because it was very easy to look for new antibiotics. So we can screen different species, such as fungi, and we can put them into antibiotic dishes. We can find out the range of chemicals that they have on offer, and so we can make a huge big library of different chemicals. And once you've got this library of chemicals, you can test all of them on bacteria. And without knowing anything about the mechanism, without knowing anything at all, we can see if there is inhibition or growth. And so there's this easy sort of mass screening technique. And then we've run out. So once you've done this so many times, there can be no more antibiotics that are natural. So what we end up having to create new antibiotics based on theory instead, and it takes a long time. I think there are actually some antibiotics since the late 1990s, but the sort of they tend to be just changing the R groups, so the variable chemical groups in general, but it's slowed down to a huge degree. To be honest, there haven't been haven't been many advancements towards making very specific results. But if you understand the microbiology, so for example, with the PVPs, let's go back to this. So I study the PVPs in my PhD, and these proteins that create crosslinks in peptidoglycan. And if you understand how they work, and you understand the 3D structure, and you understand the other elements of this. So for example, there's an enzyme here, it's called MLTB, and this actually destroys peptidoglycan. So as well as creating peptidoglycan, the system itself needs an enzyme that can destroy peptidoglycan. And so I think quite recently, there was an antibiotic called bulgicin that was discovered, and it's called bulgicin because it makes the bacteria sort of bulge, and the membranes themselves to sort of become disrupted. And so by understanding these sort of new mechanisms and how they work, we can actually do combination therapies. And I think that the real results that have been achieved recently are these advents into combination therapies where we use multiple antibiotics that synergize with each other and understand the reasoning for that synergy. I think that's sort of the most recent results. I'm just going to look at another question because I think it might take a bit too long with that answer. And then during the professor's talk, I'll also answer them in text. Delvibrio question. Does it kill the bacteria instantly? It's meant to kill them within 30 minutes. So actually, it hasn't yet killed it when it's in the in the cell wall. So we'll just go back to the Del Vibrio slide, as it's quite interesting to me. So actually, when it's at the stage of this stage here, a lot of the time the host bacteria isn't dead, but it does have nucleases and other enzymes which do have a toxic effect and kill the bacteria. At this stage, it is dead normally, but in this stage, actually, when it goes in, it repatches the hole. So it uses its own PVPs and enzymes to then repair the hole that it made. So it makes a nice little home for itself. And as well as entering, it also has enzymes that sort of break apart the structure and make it into a sphere. It's very, very interesting. I really highly recommend looking up Del Vibrio, and I think it will have a big impact in the future. And then Q&A. There won't be an assessment. Okay, so when peptidoglycan synthesis is inhibited, what process kills the bacteria? The thing that kills the bacteria is the fact there's this sort of a turbid pressure within the bacteria that is held in place by the cell wall. And also when it's trying to replicate, it can no longer replicate. So if you can't make a cell wall, you can't get any bigger. And so it's trying to do everything else. And so the pressure increases. And so a lot of the time it will lyse. A lot of the time it just can't grow anymore. So rather than killing it, it's more of an inhibiting antibiotic. And I think that's the end of my questions, but I will type the answers to all those that have been put in the Q&A and also on the chat. I'll let Professor speak and have it now. Thank you, everyone.
Okay, thank you very much, Chris. Yeah, so having listened to the internal part of bacteria and the whole cell, now I'll talk to you about the One Health concept, as well as share with you research works that we have done on some of the bacteria using the One Health concept. One Health concept is basically a holistic approach to prevention of diseases. In the past, it has been seen that a lot of people have been concentrating on reducing antimicrobial resistance on only human beings, but it has been realized that this bacterium normally can be spread from humans to animals, animals to human, human to the environment. So there must be a holistic approach to tackling antibiotic resistance. So the One Health approach has been accepted widely in order to curb resistance, since it can be transmitted through all these three places. And we all know that microbes are ubiquitous. They can be found everywhere, and also they can live on several hosts, unlike other microorganisms that are very, very specific. For instance, E. coli can be in the human, animal, and also in the environment. So it's very, very important to look at them holistically so that you can cure them, as well as their genetic materials. As these microbes move also from one host to the other, we realize that their genetic materials are also spread. So that is the reason why we are taking the One Health approach to prevent resistance in antimicrobials that we have been using so far. Please, next. Yeah, so how bacteria and their resistant genes spread. Chris mentioned about all this, but let me take you through. The bacteria is on the left, the top, antibiotics. We all use antibiotics. Some use it when even they are having viral infections. And this breed a lot of resistance in them, and through their feces or whenever they are not hygienic, they can spread it through other surfaces. When they go to the hospital, they can also spread it there through the contact surfaces, and these contact surfaces can harbor these resistant genes and give it to patients, nurses that are working around, doctors that are working. In the same vein, antibiotics are also known to be heavily dosed to animals, especially in the United States. It has been estimated that almost all the antibiotics that are produced annually, about 70% are used in animal production. So you can just imagine that overuse of antibiotics in animal production. And when these microbes get resistant, they pass through the food chain, either through slaughter or their feces in the environment that are, especially when you see the situation in developing countries, feces of animals spreading in the environment and getting into water bodies. So it can also get into our food and finally into human beings. These resistant genes can be acquired. Then I also talked to you about the One Health when in the first slide, but you realize that the environment and animal, they are the, well, I hope they saw the environment and animal, you are the largest part of the One Health system, because you can eat, human beings eat animal, and human beings also eat plants in the environment. So in my next slides, I'll be talking about the research works we have done on food and the environmental samples. One of the first one I'll be talking about is the prevalence of salmonella in dams and dugouts in Tamale. You know, dams and dugouts are largely built in Ghana to store surface water, and they are mostly untreated and also used for several things that in our homes. And salmonella, you know, the typhi, which is normally the one that causes typhoid fever, but we have another one which is non-typhi, which is now the leading cause of bacterial diarrhea worldwide, and they are estimated to cause approximately 153 million cases of gastroenteritis and 57,000 deaths globally each year, especially in developing countries. So that's why we work on this bacterium. And what we did with the dam water was to take samples of the dam water. You can see animals drinking from the dam, human beings drinking, people also using this same water to water their vegetables. So you can just see if there is bacteria there, how it will be spread throughout. So what we found in the next slide, please. What we found in the dam water, out of the 70 dam and dugouts that we fetch water from, we found out that 48 of them were contaminated with the bacterium salmonella, and also try to test their resistance to common antibiotics that we had. Chris mentioned about the antibiotics. You can see that ampicillin and chloramphenicol that we tested them to, most of the isolates from the dam were very, very highly resistant to these antibiotics. What is this telling you? It's telling you that this water that our people are drinking may be a source of resistant genes for them. When they drink it naturally without even taking penicillin or ampicillin, they may be able to build resistance to these antibiotics. And that will also mean that we'll move on to the high and more expensive antibiotics to cure common diseases that affect us. Next slide. Yeah, so we also try to look at the spread of this salmonella in the food chain through water. We all know that most of our cities now use sewage, especially in urban agriculture, to water some of these vegetables. So we try to find out the prevalence of this salmonella on cabbage that are sold on the market. That's raw one. So we took samples from the market, send them to the lab, and try to isolate and identify salmonella from them. And the results is in the next slide. Yeah, so we found out that out of the 40 cabbage samples we took from different markets of Tamale, 11 of them, that is 27.5%, were positive for salmonella. You remember that I told you we had people using this dam water and also sewage water in the cities to water vegetables. So you can see how this thing is spreading. On the right-hand side, we have the resistant pattern of these 11 isolates that we took from the cabbage. And you can see that still ampicillin is very highly resistant, followed by cefotaxime and ciprofloxacin. And when you are talking of ciprofloxacin, you know that this is one of the commonest drugs that we use here in the developing country. And if we are beginning to see high resistance in just fruits that we eat, then you can also maybe take these resistant genes from just by eating these fruits that are not well prepared. In the next slide, I'll show you the next slide. Yeah, so we also try to work on processed salad. I took you from the raw cabbage that was not processed, but this time salad that was prepared and ready to eat. We also decided to take samples from them, and this work was sponsored by the WHO AGISA project. So we took samples of this salad from several food vendors in Tamale, and took them to the lab to isolate salmonella from them. In the next slide. Yeah, so you can see that out of the 133 samples we took from different street food vendors by the roadside selling mixed salad, we found out that 70 of them were highly contaminated with salmonella. And the resistant pattern is on the right side, where you have the tetracyclines and ampicillin also a bit higher, and then amoxicillin clavulanic, which is also higher. Let me tell you that the amoxicillin clavulanic, it was developed to prevent the resistance that is being caused by the penicillin binding proteins, as Chris said. But now you can also see that there is resistance to amoxicillin clavulanic, and we must be careful about that. Please, the next slide. Yeah, not only were we interested in vegetables, but also meat. You all agree with me how meat is handled in our developing countries. I've traveled across Africa, and I've seen that how we handle meat is almost the same from Ghana, Burkina Faso, Kenya, and the rest, and how we transport it. So what we sought to do in this research work was to look at the prevalence of salmonella from the meat that is being sold from the market. So we took samples from the market, took them to the lab in the UDS Spanish lab, and we isolated salmonella from this meat. The next slide, please. Yeah, so out of 120 meat samples, 124 meat samples we took from the Tamale metropolis, we found salmonella in 37 of them, representing 30% of the meat. So people may also say that meat is will be cooked before eaten, but the issue is cross-contamination. People buy this meat from the market, take them to the kitchen, wash them, and spread the water around. So the issue of cross-contamination counts. If you buy an already contaminated meat from the market and you take it to the kitchen, you must handle it with care. If not, you can easily get this salmonella. Then we also found out that tetracycline, please then slide. Yeah, tetracycline is very, very high here. Look at it, because in the following slide, I'll tell you something about tetracycline and in the meat value chain. These are the resistance we got from the isolates that we took from the meat. Next slide, please. So we have different serotypes of salmonella, about 2,000 types of serotypes of salmonella. In this salmonella that we isolated from meat from Tamale, we try to serotype it to see the different types of serotypes that we have. We had very diverse types of serotypes, some of which were first found in Africa. And you can see that the number 6 is Kokomlemle, is a resident serotype that we have. Kokomlemle is an area in Accra, which this serotype was first found. But we also found that, yes, we have this serotype here in Tamale. The next slide. Yeah, so we also got this grant from the International Foundation for Science to work on resistant bacteria in food-producing animals. As I told you, if resistance are built in animals, it can easily get into the food chain or also the environment and finally get into human beings. So in this project, we try to look at how farmers use antibiotics and also clinically isolate E. coli from animals that they have. Sorry, yes. Clinically isolated E. coli from animals that they had so that we tested them against antibiotics. Next slide, please. Yeah, so basically we did this research work in 5 districts of the northern region. You can see from my left, the top, that we asked farmers and also saw the antibiotics they were using and documented everything. Then we further went on to take fecal samples from all these animals, the poultry, goat, sheep, pig, and cattle, to look at their resistance level to antibiotics. The next slide, please. Yeah, so these were the results we obtained from the farmers who were using antibiotics. So from this table, you can tell that 92% or 92.5% of these farmers are into the use of antibiotics. And some of them use them for growth promotion. And most of the antibiotics they use were penicillin, amoxicillin, tetracycline, and ampicillin. In my previous slides, I you saw the resistant pattern in the graph that amoxicillin and tetracycline were very highly resistant. And you can see a linkage between this use and also the resistance to ampicillin and tetracycline. The next slide. Yeah, so out of these isolates, we took 180 samples, and we isolated E. coli from almost all of them. The isolation rate was about 97%. So as for E. coli, you know, it is a normal flora, okay, in the gut. So unless, of course, the animals are taking antibiotics, that will clear some of the bacterium E. coli, we will have E. coli in a healthy animal, normal healthy animal. We have found out that, as they said that they use tetracycline a lot on their farms, animal farms, we found a very high resistance to tetracycline, which was especially in poultry, which is normal because in poultry production, we also realize that because there are many and it's capital intensive, most of the farmers wouldn't like to lose their animals. So they give these antibiotics for growth promotion and to also prevent secondary infections, so that they will, in case of any disease outbreak, they will not be hit very hard. So that is the work we have done on antimicrobial resistance in food producing animals. Then the next, which is the last one, is work we did on the environment, that is sewage samples. So this work, we took, you can see myself taking the sewage sample from Tamale. We took samples from 74 countries in the world just to look at resistant genes in the environment, just by looking at the sewage in urban centers. And through that, we were able to get some interesting results, which I'll share with you. We analyzed this through metagenomic analysis. Linda will talk about it when it comes to the identification and the rest. So out of these sewage samples that we gathered from all over the world with metagenomics, we were able to classify the various types of antimicrobials that are found in the sewage. So you can see they clustered all the continents, Africa, Asia, Europe, North America, and South America. When it comes to Africa, you can see some differences, especially in Ghana. I'm in Ghana, so let me talk more about Ghana. So in Ghana, you can see that in the environment, you find this, you find tetracycline. So you can see the trend that we saw on the tetracycline is also getting here into the environment. You have a lot of tetracycline in sewage water in Ghana. Then we also have the macrolides and aminoglycosides, which is the green one, also found in the environment. So in effect, is to tell you that we have all these classes of antibiotics just in the environment. What does that tell us? That means we are using these antibiotics and passing them either by our physics or companies that are producing antibiotics are also exudating or also pouring their exudate in the environment without proper treatment. So that is what we are finding. And of course, people who use antibiotics in poultry, in the poultry industry, also are also culpable of this. In the next slide, we'll see the resistant genes. The next slide. Then this one, they are showing the real resistant genes that were found in the environment. The real resistant genes. So you can find for instance, in Ghana, you can find the TET-W, TET-Q, TET-37. These are the genes that are in the environment. And as Chris told you, through conjugation, some of these resistant genes could be shared between bacteria. And you can also bear with me that our sewage system is not the best. So the sewage water can finally get into our streams, can get into our dams, and people drink it. So in this work we have done, it has been found out that we have very, very important resistant genes that are mainly found in Africa. Most of them are not classified. You can see from the legends that the others, some of them are not classified, but they are highly resistant genes in the environment in Africa and Asia itself, which doesn't look good. So I think we must take this One Health approach into consideration and very seriously, so that we will be able to curb antimicrobial resistance in the environment, in animals, and in human beings. So my summary, yeah, I talked about the One Health concept. I also spoke about the prevalence of salmonella in different food, raw food and ready to eat, as well as beef. I also spoke about the antimicrobial resistance in food producing animals, which are very high and also are having correlation with the type of antibiotics that are being largely used here in Tamale. Then I concluded with the resistome, that's resistant genes that we have in our sewage water in Ghana especially. Thank you for listening. I'm ready to take your questions concerning this presentation.
Oh, thank you. I think if you can read the Q&A, or if you'd like, I can read the questions that one of them has given us.
Yeah, the first one is normally the ability of bacteria to resist again is found to the body with high accumulation of antibiotics, but this happens to other bodies naturally, I mean, if they never use antibiotics. Now, what is the relation between body immune system and antimicrobial resistance? Okay, so yes, it's true. It is true that resistance can also be found intrinsically, as Chris mentioned this, in some of the microbes that we have. So it's not always that the misuse of antibiotics leads to the resistance. Some are intrinsically resistant to certain classes of antibiotics. Then now, now, what is the the relation between body immune system and antimicrobial resistance? Well, antimicrobial resistance, the fact is, if you have a resistant gene, or if you have a resistant bacterium, you have a lot of them, the body system, the immune system is not able to kill that bacteria. That bacteria still exists in the body with its resistant genes, which will continue resisting other types of antibiotics. I don't know whether you are clear with this.
I would want to mention that if anybody has any contribution, especially to the one who asked the question about the relationship of immune systems and bacterial infection, you can, just let us know by raising your hand, and we will unmute you for you to contribute to the discussion. I think we are in the last part of the MOOC 1, where Prof. Carriage's presentation on One Health would see some conversation. So I think we're already in that mode already. So if you do have any questions with regards to that, just please do let us know, and we would have you to ask that.
Yeah, just to also add more to this, the fact is we are talking now about, people are talking about threshold for bacterium to cause diseases. Yes, so when you reach that threshold that the body immune system cannot take care of, then it turns into diseases. And people are also talking about threshold, or let me say, threshold relating to standards. So for instance, let's say if you found maybe one colony of bacterium E. coli in water, and the standard says that you should get maybe about 100 of them before you will be able to consider it as dangerous. In the field of antimicrobial resistance, we are no more talking about the threshold that it will reach before, because even one single colony or one single bacterium can be resistant and can spread that resistance. So whilst people may be talking about threshold to cause diseases, those who are working on antimicrobial resistance will tell you that no matter how small the bacterium is, or the population of the bacterium is very, very important, because there could be resistance. There could be spread of these resistant genes as well. Do I have more questions?
Yes, Prof, there is a question that is asking, aside inspiring communities and getting funds to conduct this research for effective management of such cases, are the policymakers ready?
Interesting. That's fine. So at times, as a researcher, you may be discouraged along the line, but don't be discouraged. With all this research we have done, we try to get in contact with the FDA so that we disseminate these results, and also organize those along the food chain to educate them on the need to process their food well, to avoid these resistant bacteria from infecting people. But mostly when I get to the FDA, they are very, very political. So they are very, very political, and they are demanding. And interestingly, other bodies, WHO and the rest, are more interested in my data than my country. So I cannot force them. Some of the research work we do them with our salaries, and I'll still force them to take my advice. No, I will not do that. If I have the opportunity to talk about it, yes, I'll do that. But going again a step ahead to use my resources again, in fact, it's not easy. So we try to do that. So some of these fora is the opportunity for me to disseminate this. If you call me at any time, at any conference or any fora to talk about this, yes, I'm ready to do that. So that's the way we can share this.
I'm slowly letting people talk so that you can ask questions freely as well. And also, if you have anything interesting to say yourselves about AMR that might be relevant as well, as we've entered sort of that phase of the talk. Thank you, Professor. I loved your presentation.
Okay. So, yeah, please go ahead, Yusuf.
Okay, good afternoon, members. I'm in Uganda, and this side it is an afternoon. Thank you for taking us through this presentation. It was quite insightful, and it had very many captions when it comes to microbiology sessions. However, now when it comes to the settings where I am, we may not be able to use a lot of advanced technology now, meaning that we have a number of projects, but the projects which we can try to run and request for some funding may seem to be for a local community, not for an advanced community, because we have a number of projects we can suggest and which we are interested to take on, but it may not necessarily reach to the advanced technology. I don't know whether we can be considered in that fora, or it's a
Okay, Yusuf, I think, are you done asking your question? I think you are experiencing some technical.
I think Zachariah has raised the hand.
On the
Sorry, Yusuf, so if you can hear us, I think your question was about how can we develop a more flexible funding system that can help start local research. So to answer that question, maybe I think as part of the current project, we have a last set of micro grants that is supposed to support local communities and local researchers like yourself and others who are part of this to develop their own and very innovative projects that are capable of solving challenges within the AMR value chain. So that is the very first step towards overcoming this challenge. Subsequently, I mean, we are understanding that, I mean, per the interactions that are ongoing on chats right now, we see that people are talking about funding as an impediment to how they would conduct their research. So I think from our side, one of the first things we think, which is different from the granting system, is to have an open peer review system, which is being implemented by Just One Giant Lab, which would, we believe is a first step to democratizing the funding system and how it will help local researchers like yourself that have research questions to answer but are not having access to the really traditional and very bureaucratic grants systems that already exist. So I think that's the first step to it. And I think Chris has responded to that, that there is currently available €1,000 to start off. And as time progresses, while we have more resources, we believe that we would provide that funding platform, which is peer reviewed, and the best would be chosen by yourselves. And then you can communicate that to the community that we are building around antimicrobial resistance in Africa. So that's just to answer that. But my colleagues at some point in time would also add more to that. Do we have any other questions, please? I see Abdul Hakim raising his hand.
There is a question here I need to answer from James Opoku. Yes, he was talking about the fact that my work was on the northern sector of the country, or the northern part of the country. But is there any info on? Yes, a lot of researchers have also found salmonella and E. coli in food in the southern sector. Yeah, most of the, I have this work on meta-analysis of food borne diseases research papers that were published from 2008 to 2012. And you found out that most of the research work done were largely in the southern part of Ghana on this same issue. So that is why we are here in the north, and we would also like to know the situation of this prevalence in salmonella. Yeah, so a lot of work has been done on it in the south as well. Thank you, James.
Okay, I think that there is a question in the chat from Alex Hashkana, which is talking about disseminating research outcomes where potentially it could get people to know about the research that is being carried, and then also serving as an to inspire stakeholders and bring them on board, so that it can serve as an information sharing system. So I think one of the things that we are trying to do with Juggle Africa through the Africa Against Antimicrobial Resistance project is one of such, where we are building the community. So every research that is ongoing, we have a platform, the Just One Jan Lab, which I believe most of you have already signed up to. So that is one of the first steps to sharing your research outcome. However, currently there are really innovative ways of disseminating your research without having to publish in the paywall journals, which are open access journals, which most of them you don't need to pay any publication fees. So the first step, and when we have a lot of traction to most of these open access journals out there, one of them, which is focused on Africa and trying to get Africans to publish their research and making them really visible, is called the Africa Archive, which accepts publications from all African researchers, and also making them available in their local languages, knowing that there are barriers to understanding research, and one of which is language. So you can look out to some of these things, and we are going to make all of these resources available to you via the Juggle Platform Africa as a way of creating that resource and tools for you, while we are still trying to build a community on AMR, and subsequently trying to overcome the challenges that we see in regards to publication, funding, and all of that, just to mention but a few. Yes, there's a question. I see somebody's hand raised. Again, I see Seda Hasif. Sorry, Hasaf Ali. Can you please unmute and ask your question?
Hello, Doc.
Oh, Samara, yeah, Samara, you can go ahead.
Yeah, Doc. Thank you for the presentation. Doc, I want to know concerning your research on salmonella and E. coli in the northern region. I want to know what was the scope. Did they cover Upper East, Upper West, or it was just around northern region, so that we can know how serious this resistance to antibiotics are? Thank you.
Okay, thank you very much. Yeah, my work was mainly in the northern region, as I indicated on the title. So it's in the northern region, not specifically in Tamale, but also different communities, Sanarigu, Tolong Kumbungu, Savulugu, Yendi. Yeah, so those were the locations that we took our samples from.
I think what's going to be really interesting is Harry's app, Microbes. It's capable of tracking the location of strains, as well as the antibiotic resistance profile, and also identification of those strains using the API test that we mentioned before. We're going to be talking about that next week. And I think that the fact that it's open and that anyone will be able to add to it, it could be a big step in tracking AMR in these communities and seeing actually where all the AMR is. So you saw the incredible results that the professors showed us. Imagine that, but it's not the one sample. You can actually see where all the resistance is going on. And I think that that's MOOC 5, and also part of MOOC 4. We'll be talking through that and hopefully trying to publicize it, and maybe even a few months later we'll see some research outcomes from that as well.
Yeah, so I think that is well said by Chris. That's to answer the question on the role of technology in helping address AMR. So we have carefully designed the MOOC to touch on some of these very important themes. So we are going to speak to the role of technology and how we are using a novel platform that we currently developed called Microbes to enable bacterial identification, both gram-positive, gram-negative, and subsequently identifying the AMR patterns, and also digitally mapping that, where we believe that technology would help in aggregating data on antimicrobial resistance, which is currently fragmented. And that would be one of the first steps to clearly understanding the true incidence and subsequent prevalence of AMR on the continent and also locally. So that's one of the steps, and that will be done in tense when we come to MOOC 5. However, there are also a number of other MOOCs that you'll be seeing in the coming days that will talk about identification, subsequently also on policy and the roles that we can play as stewards. That will be under antimicrobial stewardship, where we are going to see Gambeli and other experts to talk about that. So I think the momentum is really gathering, and I think the first day is really interesting to know that some of your questions have already been thought about with regards to what kind of information we need to bring and then make the most out of it. So thank you guys for questions. I think we have Rita raising her hands. Please, can you please ask the question? And maybe we can try and bring this session to an end, if not. Yeah.
All right. Good afternoon. I'm Rita from Ghana. I wanted to ask Doug about his work on the salmonella. I wanted to find out whether he did something on the clonality of the salmonella type. Are they the same clones that are there, or we are having different sorts of clones into the country? Because sometimes you read articles and realize that probably the same clones that have been circulating from ages are what are still in the system. Other times you realize that other work are showing that they are having totally different sort of clones all together coming into the environment. I just wanted to know whether he did something of that sort as well.
Yeah, thank you very much, Dominic. No, so far, what we have been able to do is to just map them using their phenotypic resistance. Molecularly, we have not done any work on it yet. We still have the strains there in the lab, so that at the appropriate time, we're going to do map all of them, do phylogenetic analysis, and look at the different strains, where they are coming from, whether they are coming from the environment into food, to animals, or animals into the environment to food, but what we were able to do in the meat, the raw meat we took, you can see the serotypes, the different serotypes that we have. When I was presenting, you find out that, yeah, we have some diverse types of serotypes in the system, apart from the salmonella enterica cocomlemle that we have, which we know that is Ghana. The rest are not serotypes that are known in Ghana. And this also is as a result of Ghana. In Ghana, we have our meat or animals being brought from different types, different countries, Burkina Faso, Mali, and Niger. Yeah, so some of those inflow of animals also can also have effect on the different types of serotypes that we have in the system. So we'll be doing something to try to map them and see the most prevalent serotypes, and also how they are being distributed among the food or in the food chain. Yeah. Thank you.
Thanks a lot. I can see there's still so many questions. What I'll do is we can keep track of these questions, and I'll answer them on the MOOC online, so we don't lose any of your time. Thank you so much for attending, and thank you, Courage and Linda and Harry and Gamli and Joy and everyone who helped put us together, and also the Microbiology Society who helped support us with their grant. Next week, at the same time, we'll be talking with Linda on the identification of microbes and also the isolation of microbes. If you'd like to find out more, and also for the recording, it's all on the MOOC, and we'll send you an email next week as well. Thanks, everyone. If you'd like, we have this kind of tradition on Jogel of unmuting ourselves and putting the camera on so we can all see each other, so it feels more like it's a friendly kind of thing. We see each other's faces, and we take like a screenshot and say hello. So if you'd like to do that, you can. Otherwise, see you around. And that's sort of the end of the meeting. I hope you,
and if Chris, if anyone has any question, they can reach me for any further clarification, and I'm available to nurture the young ones in antimicrobial resistance.
Yeah, it looks like we have a lot of questions the Q&A if you still have some questions. So we'll have to follow up on them later.
Yes, for some Abdul Hakim asked, what is our fate now? Well, our fate is, well, we have it in our hands. Is education. Now we know the extent of resistance. What do? We need to do practical education to really teach the people the implications of their actions, what they are doing, and the implications of misusing antibiotics. So I believe in practical training. Believe in not just talking practical. You isolate the microbes, and you'll be able to teach them through antibiogram that yes, this microbe is resistant to this, is resistant to these antibiotics, and this is how it happens in our body. to relate it, and they will be able to understand the issues. Here is the case, some people may not even know that we even have microbes. So once we begin teaching people practically that microbes exist and they are able to relate with it, we will be able to better or ameliorate the issue of antimicrobial